Assalamu alaikum. In this second part of the recombinant DNA-based molecular techniques, I'll be talking about techniques where we use them to analyze DNA regulatory sequences and techniques related to investigating protein-protein interaction. Let's go over the first part, and here we'll talk about an enzyme that we can use to investigate transcriptional regulatory sequences. See, if you remember, we have a gene, the eukaryotic gene, and this eukaryotic gene, we have the transcription start site, right? So this is the +1 site, and then we have transcription of exons, introns, processing of or splicing of messenger RNA into the mature messenger RNA. Now, what we are concerned with right now is these non-coding sequences, these sequences that regulate the activity of the gene. Here we're talking about the promoter region, we're talking about the basic promoter region, the Tata box, and we're talking about the promoter proximal element, this sort of like operon-like sequences, or that make genes, independent genes operon-like. And then we have enhancers and we have silencers as well. So we have region sequences, elements that positively regulate gene expression, and other sequences that negatively regulate gene expression. So we're talking about these sequences. How can we investigate these sequences? How can we pinpoint a specific element that regulates gene activity? How do we know if it is a positive regulatory element or a negative regulatory element? See, scientists are smart, okay? They're always smart, and they take advantage of things, okay? Some scientists are really creative. So they were fascinated by this fly right here, the firefly that produces this beautiful light at night. And they investigated why these flies, why the bottoms of these flies fluoresce or produce light. And it turns out that it's basically this molecule right here, which is called luciferin, which is converted into oxyluciferin. This is the molecule that produces light by an enzyme known as luciferase. Okay, so that's the basic reaction that produces this light, just like green fluorescent protein and jellyfish. Okay, sort of same concept. Okay, so they took advantage of this enzyme, and what they did is the following. Right, so used recombinant DNA technology, and the purpose of this technique, which is known as luciferase reporter assay, is to study the activity of a gene at certain conditions or elucidate the function of certain regions of the promoter region. So what do we mean by a reporter? A reporter gene is basically a gene that gives us a signal, that gives us an idea of something. And here we want to know the activity of a certain region or inside a cell under certain conditions, or the importance of several regions in terms of regulating gene expression. Okay, so that's what we do. We take the promoter region, we take the regulatory regions of any gene, and we put them ahead of the luciferase gene. So now the luciferase gene is under control of the promoter region of the gene that we want to analyze. Okay, so we're not really concerned about the gene itself, the gene of interest. We are concerned about the regulatory elements that regulate, that control the expression of that gene. So we put the luciferase gene under the control of these regulatory elements. So we have a plasmid right here again, and we have the regulatory regions, the regions ahead of the transcription start site, the +1 site, and we put the luciferase gene under control of this region. And we transfect cells with this plasmid. Transfection means that we insert the plasmid inside cells, like human cells, okay, which we culture and grow in laboratories. So now we have the plasmid inside cells. Now, the proteins, the transcription factors, the regulatory proteins that control the expression of the gene of interest would control the expression of the luciferase. Now, cells are not really that smart, okay? So they don't say, oh, wait, this regulatory sequence does not belong for this gene, the luciferase gene. They don't care about that. They just care about the regulatory element itself. Can they bind to it or not? Okay. So now, if the gene of interest is, or if it is at a certain condition where it should be expressed, instead of expressing that gene, we have the luciferase gene expressed instead. Now, the more luciferase we have, of course, we add the luciferin compound or molecule to the cells. Now, if the gene of interest is highly expressed, it means that the luciferase gene would be expressed instead, of course, producing more luciferase, converting more luciferin, producing more color. So here we are reporting, okay, the more luciferin is converted, the more luciferase is produced, the stronger the color, and vice versa. That is, opposite is true. So we put cells under certain conditions, and we measure the amount of light that these cells produce. Okay, so we would say, this condition actively regulates the expression of the gene of interest according to how much luciferase is expressed. Okay, or that other condition negatively regulates the expression of the gene of interest. Okay, and this shows the results of a certain experiment, of specific experiment. Okay, and these are true results, by the way, for any particular gene. So that's what we do. Okay, so here we have the transcription start site of the luciferase gene, and this is the regulatory sequence right here. Now, of the gene of interest that we want to study. Okay, so here we have the core promoter region. This is the RNA polymerase binding site that produces the basal expression. Okay, basal means the minimal, the standard expression of a certain gene. Here we have the activating region. All right, that's a possibly regulating or regulatory sequence or element. And let's say that here we have a repressor region, and we use genetic engineering again. Okay, and that's what we do. So we take cells and we transfect them. That is, we introduce the plasmid that contains the luciferase gene, but without any promoter, nothing. This is gone. This does not exist. This means that there should be no expression of the luciferase gene. Okay, so we have the cells, we have the plasmid inside them with the luciferase gene, no promoter. So that's what we call a negative control. That's what we call, this is the product, or this is the sample where there should be zero or minimal expression of the luciferase gene. So that's a negative control. Now you see a little signal right here. Now this could be something like leakage of expression. It could be just background color or light. It could be anything like that. So this is the negative. This is the sample where there should be no luciferase, little luciferase. Now, we also have a positive control. This is a sample where the luciferase gene is under control of a really good promoter, whatever promoter that is. Really good. It's not the promoter of the gene of interest, okay? Just really good promoter. So this gives us like the maximal expression of the luciferase, and you can see that the color is it's like 100%. Okay, so this is the 100% results. That's how it should look like. So it controls everything. Now we have the promoter of the gene of interest, this whole thing right here. So that's the complete promoter, as you can see in here, and you get a specific signal. Now, it's less than the signal produced by the good promoter, really good promoter. So, you know, it just tells us that there is some expression. All right, now what we do next is genetic engineering, where we chop off parts of the promoter region. So we take this part right here, okay? Now we still have the repressor region, the activating region, and the core promoter. Now you can see that the expression is sort of similar, by the way, to the complete promoter, because we haven't really removed any regulatory part. Now you can see some differences, but this increase right here is really not that significant. Okay, so then we remove more of the repressor region. Aha, now we have an increase. We remove more of the repressor region. Again, we see an increase, and this is a real increase. Now that compared to the complete promoter, so you can see that there is a significant difference. Now, so that tells me that this region that I have just removed contains a repressing element. Okay, so that's good. Then what we do is that we remove the activating region, and you can see that there is huge drop in the expression of the luciferase gene. All right, so that tells me that there is an activating region, an element that positively regulates gene expression, the expression of the gene of interest. Okay, and this is reported by the luciferase, expression of the luciferase, which is marked by how much light is produced by these cells. Now we remove more of the activating region, and you can see a huge drop again. We remove the core promoter as well, and you can see that there is no expression, and you can compare this to the negative control. Okay, so that tells me again what certain regions in the promoter represent. If they are activating or repressing regions, if they contain enhancers or silencers or promoter positive elements, proximal promoter elements, and so on. Okay, so that's the first technique. Now, the other two techniques are really beautiful, and the idea here is to study protein-protein interaction. Now, remember that proteins do not act by themselves. Okay, what they do is that they mainly interact with other proteins in order to produce an effect on cells. Now, the first technique is known as co-immunoprecipitation, and the other technique is the yeast to hybrid system. So here is an example of how proteins can interact with each other. So we have a protein right here, and it contains two domains. One that exists at the N terminus, and the other exists at the C terminus. Now notice that at the N-terminal part, the N-terminal domain of this protein interacts with two other proteins, and these two proteins can interact with each other as well. Now, if you look at the C-terminal domain, it can interact with 4 different proteins. Now, notice that these two proteins also interact with each other, but this protein does not interact with that protein. Okay, so this protein-protein interaction is really complex. It's not that easy, but we can use co-immunoprecipitation and the yeast to hybrid system to study how proteins can interact with each other. So how do we do that? So we talked about immunoprecipitation before, and we said that we use an antibody, so immuno and antibody. We use an antibody. This antibody binds to a protein of interest and it pulls it down. We remove all of the other proteins that are soluble or not bound to the antibody, and we can take this antibody with our protein and we can study our protein by immunoblotting, SDS page, or whatever technique. So here what we're doing is co-immunoprecipitation. We're not pulling down only the protein of interest, rather we are pulling down all of the other proteins that interact with our protein of interest. Okay, so what we do is that we have cells, we open up cells, releasing their content of proteins and everything else. We're concerned with proteins. So we add an antibody, and the antibody binds to the protein of interest. So here we have the antibody. The antibody binds to the protein of interest. Now, it pulls down the protein of interest that is now bound to the antibody, as well as all of the other proteins that are bound to the protein of interest. We remove all of the other proteins that are not bound to the antibody, and we can take our sample and we can study it by immunoblotting or SDS page. Now, immunoblotting, by the way, is also known as Western blot. So we have Southern blot for DNA, we have Northern blot for RNA, and we have Western blot for proteins. Great. So before moving on to the yeast to hybrid system, let's talk about a DNA library. So a library is basically a place where we have collection of books. So whenever we want a certain book, what we do is that we go to the catalog, or we go to the net. These days we search for the book, if it exists in the library or not, and then we can check where it exists exactly, which floor, which section, which shelf, and so on. We take our book and we read it, or whatever, draw mustaches on faces or whatever. So that's a book library. So how about a DNA library? So a DNA library is basically where we have tubes containing different fragments of DNA, and they are saved in a freezer. And we have a catalog. So whenever we want to study a certain gene or a certain DNA segment, what we do is that we check this catalog, and we know where our DNA fragment of interest exists, which freezer, which shelf, which box, exactly where inside this box. We take the tube and we can study it. So that's a DNA library. And we have two types of libraries. We have a genomic DNA library and we have a CDNA library. So what's the difference, or what are the differences between these two libraries? So the genomic DNA library is basically, we start with genomic DNA, the whole DNA in a cell or group of cells, human cells, let's say. We cut the DNA using, let's say, restriction endonuclease. We have smaller fragments. We have these fragments and we recombine them. We place them, we insert them into vectors like plasmids. So each plasmid would have a certain DNA fragment inserted inside it. We have these plasmids, we insert them into bacteria, and then we have different colonies, and each colony of bacterial cells would contain a copy of a plasmid molecule with a specific fragment. So each bacterial colony would contain different DNA fragments. Now, Remember, we're taking the whole genome, meaning that we have everything. We have introns and exons, we have motor regions, we have enhancers, we have coding regions, we have non-coding regions. Okay, so we have everything inside this library. Of course, we take these bacterial colonies and we can study what fragment they specifically contain and so on, and then we can study it. Okay, so a cDNA library is cleaner, simpler. So what we have is, first we take the messenger RNA from cells, we convert them to cDNA using reverse transcriptase. Now, and we insert these cDNA molecules inside plasmids. Okay? Now, so each plasmid contains a gene. Now remember, this gene or this DNA fragment does not contain introns. It does not contain promoter regions or enhancers. It's only exons. Okay, and these exons can contain translated regions as well as untranslated regions. The starting point of the messenger RNA, the 5' end of messenger RNA, the regions that are not translated, as well as the 3' untranslated region. So we insert these recombinant DNA molecules into bacterial cells. Again, each bacterial cell would contain a specific gene. Great. Now, a difference between genomic DNA library and cDNA library is that here, if we take, we have everything in the genome inside these plasmids. Here we have genes. In the cDNA library, we have only genes. Here we have coding as well as non-coding regions, and here we have only the coding regions, the regions of DNA that are transcribed, the genes. Now, if we take human skin cells, if we take human stomach cells, epithelial cells, we have neurons, and we create a genomic DNA library, these libraries would be identical, okay, because they all have the same genome. But if we create cDNA libraries from different cells, these libraries would be different because the genes that are expressed in skin cells are different than the genes that are expressed in nerve cells. Okay, so these cDNA libraries would be completely different. Now this slide shows the similar concepts that I just talked about in terms of genomic library. We take the whole thing, we chop off the whole genome, we chop off, we cut the genomic DNA into different fragments, we insert them into plasmids and into bacteria and so on. Here we have the cDNA library is created from messenger RNA molecules that are converted to cDNA by reverse transcriptase, and these are inserted into vectors. So these fragments in the cDNA library represent active genes in a cell. Now, the yeast to hybrid system is another example of how scientists take advantage of nature, how they take advantage of other biological systems for their own purposes or advantage. So this is how it works. See, in yeast there is a regulatory element known as the UAS element, okay, which stands for upstream activating sequence. Now this UAS element is the binding site of a transcription factor known as GAL4. Now GAL4, if it binds to the UAS system, it induces expression of a gene. Now, using genetic engineering and recombinant DNA technology, we have special yeast cells that produce that have a reporter gene that is under the control of the UAS system and the GAL4 transcription factor. Now, the reporter gene is known as the Lacz gene, which is the same as the one we talked about before in bacteria, that produces the beta-galactosidase
enzyme that cleaves lactose. Okay, and I'll tell you how it is a reporter gene, meaning that it gives a signal, it gives an indication for its activity, that is, if it exists or not, and how much of it exists in a cell. All right, so the UAS system is the binding site of a transcription factor known as GAL4. If GAL4 exists, it binds to the UAS system and it produces this reporter gene. Now, the GAL4 protein consists of two domains. Okay, you have a DNA binding domain and you have an activation domain. So you have this domain right here that binds to the UAS system, and you have the other domain is the activation domain that induces, that activates, stimulates RNA polymerase to transcribe the gene that is under the control of the UAS system. In our case, it's the reporter gene. Now, the idea here is that these domains, the DNA binding domain and the activation domain, must be close to each other in order to induce transcription. If they're not, then there is no reporter gene production. So just to remind you of the concept of domains. So we can do recombinant DNA technology creating recombinant protein, any recombinant protein, by taking advantage of domain. So a domain, as I said, it is a three-dimensional structure. It's region within a protein. It folds by itself. It doesn't need, it folds independently of the rest of the protein. It is self-stabilizing. And if we cut it off from a protein, it maintains its function. Okay, so it can still interact with other molecules, whether we're talking about DNA or proteins or whatever. So we can use recombinant DNA technology to take one domain of a protein and fuse it with a domain of another protein, creating a recombinant protein. So each domain maintains, again, its structure and function. So again, the purpose of the yeast to hybrid system is to investigate if two proteins interact with each other. And this is how it's done. So recombinant DNA technology is used to create a recombinant protein composed of the DNA binding domain of the GAL transcription factor with one protein, and we create another recombinant protein that is made of the activation domain or the activating domain with another protein. Now the idea is that if these two proteins, this one right here and this one right there, if they interact with each other, then these two domains would be close to each other and there would be stimulation or induction of transcription of the reporter gene of the Lacz gene. Okay, so if they do not interact with each other, then these two domains would not be close to each other, and as a result, there would be no transcription. Now, so what we do is we create a CDNA library. And the idea here, again, is to find out if protein X, a known protein, if it interacts with other proteins. We want to identify these proteins. So we create the following. We create a recombinant plasmid. It contains the gene for protein X. And it is fused with the binding domain of the GAL4 gene or protein. So once we have transfection of this plasmid into yeast, so we insert this plasmid into yeast cells, these cells would produce a protein that contains protein X, which we know what it is, with the DNA binding domain of the GAL4 protein. And we create a library. So we have many plasmids containing unknown Ys, different genes that are inserted into the plasmid along with the GAL4 activation domain. So you would have a protein, so in a yeast cell, you would have this produced, a Y protein, unknown, with the activation domain of the GAL4 transcription factor. So you have many yeast cells. Each one of them would have this plasmid right here. So all of them would produce the known X gene fused with the DNA binding domain, and each one of them would produce a separate gene known as Y. So we can call, so these yeast cells would produce Y1, the other yeast cells would produce Y2, a third would produce Y3, and so on. So these Ys are unknown. Now, so each yeast cell would have two plasmids, two types of plasmids. One produces the X gene or the X protein, the known with the DNA binding domain, and the other plasmid would produce a protein Y, unknown, fused with the activation domain of the GAL4. Great. So if the two proteins, X and Y, whatever Y is, if they interact with each other, then you have stimulation of transcription producing beta-galactosidase. Now this beta-galactosidase would then cleave lactose, but a special molecule is used. It's not regular lactose. It is a substrate, a molecule known as X-gal. It looks like lactose, but it has a special feature. When galactosidase cleaves the molecule, it produces a product that gives a blue color. Okay? So again, if you have a yeast cell, and these yeast cells, just like bacteria, they grow into colonies. Okay, so each colony is a clone of a single cell. So if you have these yeast cells within a colony producing X, of course, the known protein, and Y, whatever Y is, if X and Y interact with each other, you have stimulation of transcription producing galactosidase. Beta-galactosidase then cleaves X-gal, and the yeast cell and the whole colony becomes blue in color. If X and Y do not interact with each other, there is no beta-galactosidase, no cleavage of the X-gal, and as a result, there is no blue color, and the colonies would look white in color. So these are the different probabilities or different types of samples that we use. So we have a control, positive control. Okay, that is, we have yeast cells producing the beta-galactosidase. Okay, with the normal GAL4 having the DNA binding domain and the activation domain together. So that would produce blue colonies because beta-galactosidase is produced, cleaving S-gal and producing this blue color. Now we have another control. Here we have yeast cells that express only the DNA binding domain. And as a result, there would be no transcription, of course. We have another sample, yeast cells producing only the activation domain, the Y with the activation domain, whatever this Y is. Again, this is negative control. We know that these cells would not produce a blue metabolite, would not become blue. Now, and then we have our experimental sample. This experimental sample contains X with Y, whatever this Y is. Okay, so we have so it's all random, by the way. So what we do is that we add the plasmid having the X, it goes into all yeast cells, and we select for these yeast cells using antibiotics, for example, just like in bacteria. Okay, and then we take a DNA library. So we have different plasmids, each one of them having a different Y gene. And we add them to yeast cells, and each yeast cell would take up one of those plasmids. So each yeast cell would have two plasmids, one producing the X gene, the known, and the also produces the Y gene or the Y protein, and each one of them would generate or produce a different Y protein. Now, if X and Y interact with each other, colonies would look blue. If they do not interact with each other, colonies would look white. Now, so you can see this plate right here with different colonies, blue and red and white. And the white colonies do express X and Y, but X and Y do not interact with each other. But the blue colonies express X and Y, and X and Y interact with each other. What we do then is that we pick these colonies and we grow them to expand them. We then open up the yeast cells, releasing the two plasmids, and we take the plasmid that contains the Y gene that produces a protein that interacts with the protein X, and we identify this gene using sequencing, PCR, all of the other techniques that we'll be talking about, immunoblotting, whatever, all of these other techniques that we have talked about and the ones that we will talk about in the next coming lectures. Allah ma'akum.